Hello, I'm Rossin Erbe and I'm going to be talking about transcriptomic forecasting over short time periods using normal ordinary differential equations. Single cell RNA-seq data is often compared to getting single freeze frames from a movie. We have this set of poorly resolved images because the data is noisy and it comes from the single moment in time when the cells were killed off for sequencing, and we're trying to put these pictures together to understand what's happening. However, this is not quite right because we don't have the same movie with single cell RNA-seq data. Each cell is not telling the same story, even if we use clustering and marker genes to narrow down to a group of cells that we believe to have the same general purpose in programs. What we really have is more like a cluster of movies, because each cell will have important diverging features. Sort of like if we had freeze frames from multiple different Batman movies, and we had to figure out what is going on. We can figure some important things out, but the disparate plots and actors are going to make things pretty confusing. We would like to get more context on the images we have if possible. Similarly, that is a very important goal with cells. Can we take the small amount of temporal information that we have across similar cells and predict their future? Can we do transcriptomic forecasting in a similar way to weather forecasting, where for a short time we can predict what a somewhat chaotic system is going to do? To accomplish this, the key is that we need at least some temporal information to train an algorithm. For now, let's ignore exactly what that will be in a real data set and assume that we have two count matrices, one from time t equals 0 and the other from t equals 1. We want to teach a neural network to predict expression levels at one time step forward. So we input at t equals 0 cell's expression data into the input nodes of the network, and then our goal is to get the t equals 1 expression levels on the other side. We're using a neural ODE instead of a standard backprop, which essentially means we're using an ODE solver to update the weights in training. Then, once we've trained the network, we can feed its output back into the input nodes, and thus recursively predict expression levels into the future. So we go from t equals 1, and then we can make the jump to t equals 2, feed that back, go to t equals 3, and so on. Of course, we propagate error on each iteration of this process, which is why these are short-term predictions we are making. And we call this network RNA Forecaster. We first tested RNA Forecaster in simulated data. The helpful thing about simulated data in this context is that we can simulate expression of a cell at many different time points, providing a ground truth for our predictions that we can't get in actual cells. We used Boole ODE to simulate single-cell expression data based on gene networks, where we input causal diagrams like the one on the left. We specify the regulatory relationships between genes, allowing us to fit a system of ordinary differential equations that can be used to simulate the data over time. This produces data like we have in the plot on the right, where the y-axis is the expression level in a single simulated cell with 10 genes, and the x-axis is simulated time. We input the first two time points to the neural network for training. Then we want to know how well the network learns to generalize its predictions to future time points. Here we have the mean squared error loss on the simulated data for the next 50 time points across 110 simulated datasets. We compare the neural ODE to a multi-layer perceptron, which is just a 5 hidden layer feed-forward neural network in this case. For the neural ODE, we see that the error increases through 50 time points, but stays reasonably accurate for most simulations. We observe substantially less accurate predictions from the multi-layer perceptron, and through the very high mean squared error values we observe, we can see that it is prone to catastrophic forgetting because we're getting very out of distribution predictions here. Such catastrophes can occur in the neural ODE as well, but they are less extreme and less common. Still, we would like to reduce them further if possible. There's an interesting feature that arises if we retrain the networks with a different initialization of stochastic gradient descent. In this example on the left, we have a single gene in a single cell, where the bold green line is the simulated expression prediction target, and the other lines are the predictions of 10 neural ODEs. Each was given exactly the same training data, but a different initialization of stochastic gradient descent. We see that they all start with nearly identical predictions, but they diverge as they try to predict into the future, because the small differences in the weights compound as the network is applied repeatedly. Some networks perform much better than others on this example, but their performance varies across cells. So we have taken advantage of this by ensembling the networks. We train multiple networks with different gradient descent initialization and then take their median prediction. This both improves overall prediction accuracy and also prevents many catastrophic predictions. On the right, we compared a single neural ODE to 10 and 25 network ensembles, and see that while the predictions start off very close in terms of their mean squared error, as we progress through the simulated time points, the ensembles perform much better and are less likely to undergo catastrophe. So then the question is, how do we apply this data to actual cells? One way is spliced and unspliced counts similar to RNA velocity. We are somewhat limited there because spliced and unspliced counts form very different distributions, meaning we cannot use them to do high fidelity recursive predictions. Better would be metabolic labeling data. There you're doing single cell transcriptomics, but you treat the cells with modified uridine for a set time point prior to harvesting the cells for sequencing. That means you know if the transcripts you sequence were from that labeling period, or if they were transcribed earlier using the presence or absence of the modified uridine as a marker. This can give us 2 time points, expression before and after the labeling period, which has the benefit of being measured in real time. The input matrix is the unlabeled counts, plus the estimated degraded transcripts from the labeling period, and the t=1 matrix is the labeled plus the unlabeled counts. But how do we validate that our method is working in real data, where we can't generate time series of expression in the same cell, and we can't have that ideal ground truth? One way is to use a well-known biological system, such as the cell cycle, where we roughly know the expression changes needed to continue through the stages of cell division. We used SCEU-seq data from RPE cells, an immortalized human retinal cell line that divides constantly in vitro. We trained our forecaster on the 405 cells that were labeled for 1 hour prior to harvesting for sequencing. We then predicted the future expression state of each cell for 72 hours using an ensemble of 10 networks. We score the cell cycle state quantitatively using a program called Tricycle, which projects the expression data input onto a validated embedding of the cell cycle between 0 and 2 pi. This allows us to plot the results on a circle, as shown here for the 405 cells in the data that we use for training. The goal is then to use RNA Forecaster to predict the future cell states and see if the cell cycle scores follow the expected order of the cell cycle. In the plot on the left here, we have the y-axis as a metric of how well the tricycle scores we get from RNA Forecaster's predictions conform to the order of the cell cycle, where the scores are awarded for being both in the order of the cell cycle and not making unreasonably large jumps for a 1 hour increment. To clarify, RNA Forecaster predicts the expression levels at each hour. Then Tricycle is applied to those predictions to determine where in the cell cycle that prediction lies. Then we're applying this metric to determine the fidelity of the predictions to the cell cycle. We see here that the ordering of these estimates does decrease over time as we go from day 1 to day 2 to day 3, which we expect due to error propagation. However, even at day 3, the order is still significantly higher than random sets of tricycle scores. So the order of the cell cycle scores is quite well aligned with expectation. We can look at these cell cycle estimates on a circle that represents the cell cycle, where we have all cells starting in G2M, and we're looking at how they progress. What we see is generally steady progression along the path of the cell cycle, some waffling, but generally moving in the right direction. However, you might notice that since each frame is an hour, this is not moving as quickly as we might expect for cells that usually divide once every 24 hours. So the major limitation we face here is that we're not capturing the faster cell stage transitions, and we instead follow more of a smooth trajectory through the cell cycle. We see this clearly in a 1-cell example on the right. Here the cell slowly increases in score over time, comes back around to the beginning of the cell cycle, and starts going up again, which is very high fidelity tracking of the direction of the movement of cell cycle genes. But it's going too slow because it hasn't even quite come around for a full cycle in 72 hours. RNA Forecaster seems like it's not quite grasping the phenomenon of the cell cycle checkpoint and the ensuing more rapid changes in gene expression. It's possible that the small number of cells we have for training is the source of this issue, or we might need some change to the model architecture to be able to account for the more sudden changes in expression that we expect to see after cell cycle checkpoints. However, we think that the high fidelity to the order of the cell cycle we observe, even if the timescale is somewhat off, does demonstrate the ability to forecast future expression states in biological data. So to conclude, RNA Forecaster serves as a proof of principle that we can predict relevant information about future expression states in a cell over short time periods. We avoid some pitfalls of recursively applying a neural network in a very high-dimensional setting, using network ensembles to reduce catastrophic forgetting. RNA Forecaster is generally successful at predicting expression levels, but may encounter difficulties with more rare regulatory events. Dealing with these cases may require additional network architecture to resolve. Attention modules seem promising for this purpose. And that brings us to the end. Thank you for listening.